Hi, everyone. So today I'm going to be talking to you about a project I've been leading in the Berg Lab at the University of Chicago. I'm a postdoc there. The topic, just to give you a heads up, is going to be leaning more towards human data, but the problem is sort of has some generality, right? So from the top, as Nick in his talk was talking, a lot of traits that we're interested in studying, whether, human perspective of clinical interest, or even if we want to study them from some evolutionary perspective, are complex and highly polygenic. And the way we simplify the problem is by just imagining that the phenotype is determined by the effects of the genes and effect of this multifaceted factor, which is the environment, right? And we're very interested in trying to capture genetic effects on phenotypes. We want to understand, how our phenotypic difference is generated from genetic variability. And one tool that we have in our arsenal is a GWAS, the genome-wide association study. We're taking advantage of the fact that now we can sequence at the site level at a large number of ostensibly unrelated individuals. And we fit this simple model at the site level to ask, what's the effect that a genotype has on the phenotype that I'm interested in? And we also, after that step, get the effect sizes of at the genome-wide level. We ask the question of which ones actually matter for that trait. We ascertain these sites based on some p-value that we can decide on. But the idea is that we're picking sites that explain a certain amount of heritability in the trait, right? The cool thing about this study is opened up the possibility to use other tools based on these effect sizes that we've estimated for a given trait. One cool aspect of it, in my opinion, is the fact that you can go to a completely different database where you might have no phenotypic measurements on individuals, but you have info on their genotypes, and you use the effect sizes that you've estimated to build an aggregated score, a polygenic score for that trait, right? That opens up avenues to ask questions without actually, measuring phenotypes, because we can ask questions on the polygenic score aspect and ask, does it covary with some covariate of interest, some environment? And we can ask questions either from a clinical perspective, as I mentioned, or even evolutionary aspects. All right, so an issue that arises here in genetic data sets, I'm giving an example of humans, but this also arises in other species, but it is the idea of population structure that was just mentioned, right? On the top left here, I'm showing a PCA plot of a European database. You can see that the ostensibly unrelated individuals are actually clustering together because a lot of people share genetic ancestry, and that makes them cluster together. So we have this structure in our data set. And what ends up happening, it creates an issue when we look at polygenic scores and ancestry, and we're trying to ask some questions about the genetic signal of certain traits. Here I'm showing you on the left is the purple dots from the giant database. When we use the effect sizes from that database and look at the polygenic height score, is telling us, hey, there's an interesting story here. There's a genetic signal of this polygenic height score with some environmental or geographical gradients. But when we look at the same thing in the UK Biobank database, so effect size is estimated using a wholly different database, for the same trait, we find no signal at all. And the reason, in a subsequent paper from Sohail in 2019, showed that the reason why that signal appeared in the giant database is because of some non-random systemic correlation that exists between the effect sizes that were estimated and the allele frequency differences across different ancestries. So you had an environmental covariate that was generating a fake genetic signal. So how do we deal with this problem? The problem of confounders that exists in these data sets is the fact that you can have some environmental factor that affects your phenotype. It's hard to measure it, it's hard to include it, and it correlates with some axes of ancestry. What that does is creates this false genetic signal, so we deal with it by modeling the ancestry itself. We usually use it using PCA. We use PCA on genetic data sets. It outputs sample PCs. We include them in our linear model, and we hopefully are controlling for the bias that generated in the genetic effect sizes. However, the question that arises, which is very difficult to study empirically, is what happens if the PCs, the sample PCs that PCA gave you, are actually inaccurate to some degree? They contain some statistical error. How do those affect your estimates? Because you're hoping that the PCs are going to protect you from some bias generated in the effect sizes. And so here arises the issue of residual stratification bias, and hopefully I'll show you how that arises, because we're going to use some theoretical modeling. That's why I'm presenting in this conference. And the idea is we're going to use some math to try to understand how do we model PCA's ability to control for stratification bias in the context of a GWAS phenotypic model. All right, I'm going to start by using a very simple toy model to guide some intuition, build a heuristic argument together. This is the phenotypic generative model that we're going to be using. We're going to have some phenotype Y affected by, the genetics of some site, and crucially, the effect here of this vector U. Vector U here is just telling us individuals could belong either to population A and population B. And population A and B are related to each other using this simple demographic model. There's a split between them at some time t in the past, and so we can use the measure FST that was mentioned just in the talk before me as a measure of their genetic differentiation. But crucially, at a phenotypic level, the fact that you belong to either populations has a phenotypic effect on your trait. So individual from population A and population B can have around phenotypic magnitude difference of 2 gamma. Right, so this vector U is the confounding axis. And we're going to fit a GWAS model. We don't have an idea, the fact, a priori about the data, how these individuals fit to each population. So we're going to use an estimate that hopefully fits well to this axis that separates these individuals. And we can control for the fact that they're confounded phenotypically because of this unknown confounder. All right, so where does the math come in? The math comes in the fact that we want to be able to model how does PCA control for stratification bias. Patterson et al. In their paper in 2006 were interested in the question of when or how big the database has to be able to find the signal of structure. In this case, this FST, when did population A and B separate from each other in the past? And they use random matrix theory asymptotic results that tell us that PCA in high dimensional data, for a given eigenvalue associated with the signal you want to capture, it has to be larger than that equation on the right, the 1 plus the square root of the ratio of the sample size to the number of independent sites L that you're using in the sample. If you assume a 50-50 mixture between the two, which is what they do, you can derive what that eigenvalue, the theoretical eigenvalue associated with the signal is. It would be 1 plus n times FST. And so they solve for what is the FST need to be, or the sample size and number of SNPs you need to be able to detect a given signal of structure in your genetic data sets. And they find the solution. We're more interested as well in the eigenvector behavior. Right? We're not just the signal, the eigenvalue, but the eigenvector is associated with it because it's going to separate the individuals in our model. And McVean used in his paper relating PCA to a coalescent framework showed that as you add more independent SNPs, the consistency of your PC in capturing, recapitulating the separation between individuals, population A and B, becomes better and better and better, right? This is as a function of L. We're more interested in the function of the sample size and as well details about the eigenvalue itself or the demographic model underlying the phenotypic model. And so this is the results from random matrix theory around the behavior of the sample PC. So the B squared here is just telling us an idea of the correlation between the sample PC that PCA ought to give you and the theoretical population PC that you want to capture, which is the separation between A and B based on this demographic model. And it's also a function of the sample size, the independent sites, and the size of the signal of structure. Right? This is in this simple toy model. If we model it numerically, you can see what happens. This jump that you're seeing in line orange and blue, this is the phase change in the behavior of the detection of the signal. You have enough of a size of a GWAS now that you can detect the signal, and then the correlation is almost perfect, right? The black one, when the size of the magnitude of the signal is large enough, you have no problem at all, even at a GWAS sample size of 100 based on these asymptotic results. Good, so in this very simple toy model, you wouldn't have that much trouble because you're going to be able to detect the signal and be able to recapitulate the theoretical PC very accurately. But we're going to go to try and model reality, which is there's no two-population model that describes the reality of genetic data sets. But at the same time, the messiness of the demographic model that might underlie reality is also very, very messy. We don't know how many axes of structure there are, all the magnitudes of different things. It's hard to model it and capture it mathematically. So what we're going to do is just say that we're going to move one step along the kind of complexity axis and say that our confounded axis between A and B is embedded in some more complex scheme where now you have this older split of some population C that split from the ancestor of both A and B, right? So now we're going to use as our device the 3 population split model to study the issue of confounding bias. Same assumptions, it's a balanced mixture between A and B, and we're introducing this parameter theta that is telling us how much of the confounded individuals, A and B, make up of the total sample that PCA is operating in, and you're estimating effect sizes. And so if we use McVean's approach to kind of find what that theoretical eigenvalue associated with the same confounded axes that we were finding before, but now it's in a different demographic model, it would equal 1 plus theta n FST2, right? So now your FST2 for the same sample size as the example before, now there's the scaling factor theta. So what that means is if you're operating the same sample size, same number of independent sites, and you assume this demographic model, your PCA will fail to detect that signal. So you need to modulate some of these things. You need to add more individuals, or you need to change the sample heterogeneity in your sample. Interesting. So what that means is the same thing for the correlation of the eigenvectors, right? Now, the correlation of the eigenvectors is dependent also on theta up to the order of theta squared, sorry. And we're doing the same thing. We're trying to separate A and B so we can control for the confounding bias. And this is how B squared now looks like. What I want, the solid lines are for a small 10,000 sample size, and it grows with different line types, right? And different colors are different magnitudes of signal structure. So even at numbers of like the UK Biobank release of number of included individuals and number of independent sites, you can see that the problem arises, especially when theta gets smaller and smaller, meaning the fraction that A and B make up of your sample becomes smaller and smaller. And the reason is that for that is that PCA has less information from individuals to actually detect the signal. And that's especially crucial because this is how the issue of residual stratification bias arises, is that instead of being able to perfectly recapitulate your theoretical PC, you're in this gray area in between where your sample PC is kind of inaccurate. It's capturing something, but not completely, and it could leave some problems. So how does that arise in the phenotypic model? Same thing, same phenotypic generative model, but we're assuming a different, same phenotypic generative model, but we're assuming a different demographic model now. And so now we're kind of capturing this, we're going to use the second sample PC that PCA gives us, right? And now we've seen from that heuristic argument before that it might have some inaccuracy in it. From the GWAS model, we can express what the estimated effect size would be as a function of the true causal effect, plus epsilon here, which is due to sampling noise, and then the delta, which is the thing we're interested in, which is the confounding bias component of the estimated effect size. And under the assumptions of this model, we can find an expression of what the square root mean of this bias is. It would be a function of the absolute value of the effect that the confounder has on the phenotype, the square root of theta FST2. This represents the amount of variance as explained by the second axis that we have in this demographic model, which is our confounded axis. And the 1 minus b squared there is the error in the estimated PC. That's the degree of stratification control. So how does these look like numerically? So we'll assume that for now that gamma is 1 to make things easy. So we have on the left side the weight. This is the proportion of variance explained by the site. It increases as theta increases because there's more A and B individuals in the sample. And you can see that the error in the PC, as you increase theta, the error goes down as a function of other parameters as well, like the signal of the magnitude of structure and the sample size. What that creates when we put them all together in terms of the bias is that it creates a non-monotonic behavior. So what's happening here, to help orient you, is if we look at this left side here for n of 10,000, what's happening is as you add more individuals, A and B, of the confounded axes, the bias is increasing. The bias component of the effect size is increasing in magnitude, up to a point where now the PCA can kick in based on the asymptotic results, and it drives the bias back down as you add more and more individuals. The height of the bias, or the magnitude it can reach, depends on the sample size, but you can see even in UK Biobank numbers, the bias is non-trivial, and it depends on what's the strength of the structure that you're dealing with in the demographic model. Same thing in the sample, if we look at as a function of sample size, also an interesting behavior that is determined by this PCA behavior. The problem is also worse in meta-analyses, because in a meta-analysis, what we're doing is we kind of trying to estimate effect sizes based on different cohorts that we collated all together, but the PCs themselves were estimated in each individual cohort. So even though your GWAS is applied to a sample size n, the PCs were estimated with sample size m, which could be much smaller. So you can see in this example here on the left that if you have 100 cohorts made up each one of 1,000, and we, for simplicity, assume that they're all identical to each other in their demographic model and other details, then the problem really blows up in terms of the magnitude of the bias that we expect in the effect sizes. And so what happens if we decide to take these effect sizes and we're going to use them for a polygenic score? How do these biased effect sizes influence polygenic score results? And so we can take a sample of individuals made up of A and B. For simplicity, we'll say that the test sample is made up just purely of A and B individuals. And then we know that the contribution of each site to the bias in the mean polygenic score differences between individuals A and B is going to be a function of the magnitude, the square root of theta at FST2 times the stratification control. So you can see that the stratification control by PCA shows up here also in the bias and the polygenic score. We do some conditioning to kind of standardize our measure, and we take into consideration the fact that we're ascertaining the sites. So we're just going to, for simplicity, assume that we're taking sites just at the ascertainment threshold. And then we find that the expected per site bias in the polygenic score, standardized by the under neutral expectation, would look like something like this expression here on the right. Right? If we plot this, and we assume, for example, that we have a trait, and we're using the canonical p-value of 5 times 10 to the minus 8, and we happen to include only 100 sites, you can see the non-monotonic behavior also appearing in the bias and the polygenic score. It increases as theta increases because you're including more individuals that are biasing the effect sizes, but at a certain point, the control kicks in and it reduces the bias. The problem is even worse if we decide for some reason that I'm interested in a trait, and I need to relax my threshold to include more sites that explain heritability. So we include 1,000 sites. That blows up the bias even more in the polygenic score. So you can see the issue in that certain decisions that you might make might include more biases in your effect sizes and bias these polygenic score tests. So what are the takeaways from all this so far? Is that this problem of residual confounding under just this device of 3 population model is worse when the FST, or the signal of structure along the confounded axes, is small. So fine structure might make things worse. Or if the sample size that you're working in is small, you can think of, if we're working with ancestral DNA, the sample size might be small, and we have to control for confounding in those as well. The bias, as I've shown, is a non-monotonic function of the sample fraction, or the eigenvalue that we're trying to estimate. And the problem is even worse in meta-analyses. The fact that you have to estimate the PCs that you're using as controlling covariates in single cohorts, but you're going to use effect sizes estimated in the whole total sample. So that might create an issue. And finally, most importantly, that even in UK Biobank numbers, there is some risk of polygenic score confounding that we need to consider. And especially for cell axis of structure, and there's more and more individuals who are interested in studying, within the UK, for example, how use polygenic scores to study certain traits. And so at that level of structure, you could have some bias that could affect your results. I haven't shown some more results, but polygenic score issues are worse also for smaller samples in the meta-analyses, and if we include polygenic scores for more polygenic traits. That's about all I have for you guys. I want to thank my lab. This work would not be possible without them, and the Department of Human Genetics. I'm grateful for all of them. I want to also thank the PCB group, PopGen Supergroup, and all of you for listening and the opportunity to talk here. Thank you.
All right, let's take a question back there.
Oh, very cool. This is a stupid question, but I was trying to understand why it's stupid. So if you have this, so you showed that you have this populations where the bias was low if you were just dealing with a pair of population for a decent size. What if you did this pair by pair for all populations that you have and try to sort of take some sort of average or some sort of things across this pairwise PCs and effect sizes?
So you run PCs with using pairwise populations?
Yeah, so A, B, AC, and B.
It's a good point, but then you trade off the fact that you have to work in a much smaller sample size, and that trade-offs behavior.
But if there is enough samples, then that is a possible.
Yeah.
I see.
Yeah. But this is why I believe this project is important, is that it's trying to just to look at different parameter ranges where things might become interesting in terms of biased effect sizes.
Thanks.
One more question. So, excuse me.
Hey, really cool talk.
Thank you.
You were focusing on the effect of this kind of like outgroup in your sample. Do you have a sense of what would happen if instead you added like an A and A prime, like substructure in the in groups you're trying to differentiate?
A very good question. That would be a next direction, but in a general sense, that would be the goal, because we don't want to rely on this device. We want to be able to make general statements, which is what we're heading into right now, which will allow us at least to make statements about things like what you're proposing. Like you might have a priori some idea, and then you could think about how PCA might behave under these demographic history assumptions.
All right, let's thank Walid once
Thank
again.
you.